So if I've received your mail, why is the hybridization buffer so expensive?
Oh, my mic was off. I'm sorry. So most of the reagents are not that expensive, except of those this hybridization buffer.
Does it contain gold or what?
Silver and platinum particles. I'm joking. Yeah. And don't so 33, 34, and 35 are from a company called ACD. This is so-called single molecule RNA in situ hybridization. So you can see that it is also very expensive, but we are not talking about this in this course. We will talk in next year, probably.
So the RNA inhibitors are very expensive as well. But what is the amount? I mean, for how long can you use it?
I really don't know. I should see. So you only use 1 to 2 microliters. I don't know what is the bulk quantity. Let me check. So Part just call me. He's going to be late with 10 to 15 minutes. So please, we're going to wait. I'm sorry, but yeah. German buses are not that accurate. I was checking the RNAs. It's, how much is it? So the people, hello, to the people that are joining now, we will be late with 15 minutes. So I have searched for the RNA that you are talking about. 1 microliter contains 20 to 40 units, and we use only 1 microliter. The total amount in one Eppendorf tube that is provided is 1000 units. So this is around, this is for a lot, basically. I need to calculate. So for for those that are joining now, part is going to be late with 10 minutes, so we will start in 10 to 15 minutes. I'm sorry for the delay. Dr. Hachmarian, I'm sending you now an email with the books because I'm not coming to the hospital today.
Hi, thank you. I will share it with the other colleagues. I will now share you my email or probably on the genetic department. And sorry for not speaking on the mic because I have 2 children around. So that's why I am never answering on time.
No.
A nice head, by the way, David. Okay, thank you. It's really nice. I'm sorry for my stupid questions. I'm supposed probably to know much more about the methodology, but we actually are not dealing a lot with this. And I'm a little bit ashamed to tell you the truth, to ask you all these very basic things, but I'm really into I wish to implement some new techniques in the laboratory. So thank you once again.
No problem. So I was in Germany 2 times. I was taught by Bart and Murat about the ISH techniques. Now I'm doing ISH on monkey brains in the hippocampal region and in the subventricular zone region. So we have done RNA sequencing and we have discovered some genes that are very interesting. And we are searching for some neuronal stem cell genes that will be relevant to our studies. We have found one very cool receptor, apelin receptor, that is expressed in the human brain also. I can show you pictures. We have some very nice photos. Yeah, just studying neuronal stem cells. And also because apelin has a ligand to it, I will show you photos. They are very beautiful. So I hope that you can see. This is the lateral ventricle of the human brain. And this is the ependymal layer. And beneath it, there is a gap region without cells or with little cells, or yeah, numbers of cells. And you can see here, this beautiful cell expressed the ligand for apelin receptor. Also, if you have time, you can take a look at the database that is created for the genes that are expressed in the monkey post ischemia. So you can see here, this is the site. It's with collaboration with Max Planck Institute in Göttingen and Medical University Varna with the help of Humboldt Foundation, because Professor Anton Tonchev is a Humboldtian. And you can see here, if you click browse on genes, you can see all the 150 genes that are done on the monkey brain. And you can see here the apelin receptor. Oops. You can see also the sequence and reverse and forward primers, which is very nice to have those. So when you're want to do some experiments, you know your gene, you can first search some databases like this one or GenePaint for the primers. If you don't find the primers, you can design them afterwards. And here are some photos. Oh, I cannot load the photos. I'm sorry, because of my internet. But you can compare the control and the ischemia, which is very nice if you click this button here. Yeah, I will send you the link now. Oops. Should be clear. David, can you open the link and share the pictures, because my internet doesn't allow the pictures?
I'll try my best.
You can click on Broad Genes. Upper right corner. You can click on whatever gene you like. On the photo of the gene. Yes.
Martin, is the picture that you have shown us made by the microscope in your department or in Germany?
Yes.
Wow.
No, it is done here.
Is that
Everything.
I have opened it, but it's much uglier than yours.
Again, please.
No, I have opened the pictures. I can share with you, but it's not such an amazing one like yours.
No, those are just to see the expression in the. So this is David will show us. This is the nucleus caudatus, and the ventricle, and the important part is between the ventricle and the part of nucleus caudatus, the subventricular zone where neuronal stem cells reside.
Is that what we should see?
Yeah.
How do you analyze it? What is the expression? I can't.
If you zoom in. No, wait, I will show you. Wait a second. On Gene Paint, I will share also the link. There's a very good explanation about the expression patterns that you are going to observe.
But still, it's genetics is much more going to not to DNA sequencing, but to RNA. Physiological. Yes, I just missed the word now. But anyway, I don't have a nice idea how can we use it for diagnostic purposes, except for science at this moment. And I'm always thinking if we can implement something more in the practice. And I will appreciate, because you are more into this topic, if you have any papers or anything else where this knowledge is applicable to clinical diagnosis of genetic disorders or something like this. And by the way, I'm really not very aware of CRISPR techniques, and I'm not sure how difficult it is, but they say it's pretty easy. And I think that this paper that we have found is very interesting, because they say this technique can be done for 15 minutes, and it gives much information about expression. I don't know if you were able to read it, and do you think we can do something like this? It sounds very fancy, by the way, also, so maybe they will fund us if we decide to do it.
The best person to ask is Dr. Eugo. We will have a meeting with him today, late in the afternoon, because he now works in a company that's I don't know what exactly his work is, but he works in a very nice company. So he's the person that will translate the ISH to the clinic. And
great.
tell you all the information you need.
Okay.
I have mailed the question you have yesterday with the article, so I hope that he will read it and answer your questions. About the expression, I forgot. So you can see here 3 types of expression. We have weak, medium, and strong. Weak can be also the expressions can be categorized upon the number of cells that you observe. So depending on the, I will show you. I cannot share, but you can see the difference.
But is there an artificial intelligence that can tell you, or you have to do it by yourself?
The thing is that you have to do it by yourself. In the university now with Associate Professor Pavlov, we are working on an algorithm which can tell us the expression automatically by AI. But we're in the beginning. There is a software also, but they are not very accurate to tell the expression. Like Cell Detect is one of those softwares. And for example, every time you stain tissue, you should do a control, one without the probe, because this weak looks very like there is no staining. You can see here are some cells, but the weak ones look exactly as the controls from time to time. So it's very difficult to quantify the weak ones. The strong ones are easy. You can see the cells. The weak ones are very hard to do. So while we're waiting, I will tell you what we are going to do today, what we did yesterday. So he will be here soon. So today's program, with a little bit of delay, of course, we will show you the second part of the ISH protocol, the staining protocol. Today we are going to stringency wash the slides, and we are going to develop the signal. Yesterday we did I'll show you the protocols. By the way, the protocols are already in the site. I will send you the link. We have also included a probe preparation file. So and the slides were not with amplification, which is very important. So we permeabilize the tissues, we wash them, we apply the protein escape, Of note here that different tissue require different protein scale amounts. And we hybridize the tissues with the probes. So for the preparation of the probes, part is here already. So for the preparation of the probes, you need a file that we have provided. Here is the file. Come on. I don't know my credentials in medical university. There you go. So for working concentration of 500, we are going to use Hive mix that is 0.25 milliliters, very important, milliliters, not microliters, 2.5 microliters of tRNA and 1.25 microliters of probe for the working concentration of 500. Hello, Part.
Hello, sorry, I am a bit late. Okay. Okay. So now, so you can hear me, right?
Yes, my mic was mute time, sir.
Okay. So once the hybridization is complete, so we will take out the slides from the oven. And we have the sodium citrate buffer. So that is 2 times sodium citrate buffer, and we have to do the washing steps. So we will use 2 types of buffers. The first one is two times sodium citrate buffer, and the second one is 0.2 times sodium citrate buffer. So this stringency washes are very important. This we have to do properly so that we get very less background. So the first wash is with 2 times sodium citrate buffer, and so I hope I can zoom in. Yeah. So these are the slides, and I will first remove the parafilm. So we will again use these small containers. So we will put the slides in here. Okay. So this wash has to be done at 60 degrees. So the first time we do it at so we do this wash at a higher temperature, and then the next wash will be at room temperature. So I will keep my slides at 60 degrees now.
So the purpose of this wash is to wash the unbound RNAs molecules. And of course, to prevent the background staining. So if there are some RNAs that are not bound to the tissue, they eventually will be stained by the precipitates that we're going to use. So it is important to remove those unbound RNAs from there.
Okay, so what we can do now in order to save time is there is a 0.2 times sodium citrate wash, and that is for half an hour. So 2 times each. So we can do only one wash here, and we will do it at 60 degrees as it is. So like we have to. So basically, it's recommended that you do 2 washes, but now since we are late or I am late, so I will reduce it to like 1 wash. Okay.
Yeah.
And yeah, the other things remain the same. So if so we have like this 90 minutes incubation today with the anti-deoxygenin, the secondary antibody. So maybe we should perhaps do this before lunch.
Yeah, and we can proceed after that.
Yes, exactly.
I think every blocking buffer can be used.
No. Hold on. So he's asking, can we use donkey instead of sheep? Kind of, no, not because the anti-dig which we have that is prepared in sheep. So
Ah,
we cannot. So you have to be careful. So if your anti-dig is prepared in donkey, then you can use donkey serum, of course. But what I have from Sigma that is prepared in sheep.
I
So
didn't know.
So at the end of the protocol, here the last step we have the staining, that is TMN plus the substrate, the NBT and the BCIP. So this usually I know that it will take more than an hour. So what we can do instead is I have the slides which are stained with TNS3 and also NKX 2.1, so I can show you the results so that you don't have to wait for 1 or 2 hours, until the signal develops, so that you get an idea of how the stain looks like. Yeah. And basically on the next day, okay, Martin, if you can scroll down, please. Okay, so the next day it's just washing normally with PBS, and then you do the cover slipping, which you use the hydro mount. So that is very very simple. So, and after you do the cover slipping, it's better to dry the slides in an incubator at 37 degrees. Uh, so the sections I have used, they are 20 microns thick. Yes, the
Yes.
cryo sections. So basically, I have used sections which are 10 microns, 15 microns, 20 microns, 25 microns, and all of them have worked fine with this protocol. So basically, if you have very thin sections, then you have to be careful with the amount of protein escape concentration. Yeah. And that, like, so the advantage of thinner sections is that you will be able to see individual cells very clearly. And also, when we are in the PBSD steps, so there is a twin 20 solution. So you have to be careful that you only use twin 20 and not Triton, because Triton is generally much more harsh than twin. So we have to use a mild detergent here, not a very harsh one.
What about that though?
What? 0.2x?
Is it
About
is this the concentration?
yes.
Yes. So what I generally do is that I prepare a 20x stock solution of sodium citrate. So you can prepare a stock solution of 5x, 10x, or 20x, and then you can dilute.
So what is the purpose of dilution then? So you started with 2x and then you go.
Yeah, yeah. So basically, after the probe hybridization, what we are doing is uh, uh, a strong stringency wash. That's with a concentrated citrate buffer. Then in the next step, we do a lower stringency wash. So we have to use two different concentrations. It's just to remove the unwanted probe, as Martin mentioned. So the main idea of the washes, you can also increase the washes. Like if you feel that there's a high background, let's say, so then you can always play around with the washing steps, and you can increase the number of washes and try to reduce the background. Do you neutralize the pH afterwards?
So what is the pH of the citric buffer? Is it acidic, right?
Yes, so I should let me check.
I don't think at this stage the pH is that important.
No. It is important actually. Maybe yeah, we should write the pH of all the solutions. So yeah, so citrate is acidic, and then we normalize the pH around 7.5.
So the stringency is actually meant to, by using this acidic pH, to detach the RNA probes that are, let's say, unspecifically bound to the tissue, right?
Yes, right.
And then you go to the pH of the PBS. In this case, you have added tween 20, right? And then you continue at neutral pH.
Yeah. Okay. So now I took out the slides from 60 degrees, and we proceed towards the next wash. That is again with 2 times sodium citrate buffer, but only for 5 minutes and at room temperature. If you want, you can also do the washing steps on a shaker at a very slow speed, but generally it's not necessary. But if you want, you can also do that. Okay, so now we will proceed towards our third wash. So with 0.2 times sodium citrate buffer. So as I mentioned, we will do this one time for half an hour at 60 degrees. So are there more questions so far regarding anything, the primer design or template or probe?
There is a question from David. What's your recommendation? How many samples maximum should one do at one go to still have nice results?
Yes, so I would recommend not to handle more than like 20 slides at once. Like if you have this, so if you have these big containers, these ones, so here you can easily fit 20 slides. And you can handle 20 slides at once so that it's easy to wash and do, but not more than 20. And yes, you can do sequential staining. For example, you can, so there is a washing step with TMN. So the second last step, you can, let's say, store 10 slides in TMN and proceed with other 10 for staining. Once, so you can store the slides in TMN for some time, but not like for a couple of hours. No, for, let's say, for 1 hour, it should be fine. It will not harm. Also, during sectioning, you can, for example, fit more sections on one slide. So let's say all our mouse sections, so we have 4 sections on one slide. You can also have 2 sections on one slide or 1 section per slide. So yeah, so what Martin is showing, so this is how we have the slides.
Because in the institute, they have, how is it called in English? Workshop, meta workshop. And they can design all those goods. So for example, this is a chamber. Mark, can you explain this chamber?
Yes,
What's
so
the purpose?
yeah, so this is a cryopreserving chamber. Basically, so when we dissect any tissue, for example, brain or the mouse embryo, so we have this a copper plate which is brown, right? So we fill this chamber with the freezing medium. So we have the OCT freezing medium, and we fill this 3/4, and then we put our tissue in here in such a way that the tissue is exactly placed at the center from all the sides. So it should be
Thank you.
exactly in the center. So as you can, I don't know if you can see, but there's a plus sign on the in the chamber. So this helps us to orient our tissue that it's exactly in the center with equal distance from all the four sides. Yeah, and then we place this whole chamber in a uh, in a ethanol box, like, which is at -80. So then gradually the freezing medium, it will freeze from bottom to top. For very soft tissues, for example, kidney or liver, it's not recommended to use this freezing chamber. Then you can do paraffin sections. So this is the gene pin database, and if you want to look at the expression pattern for any gene in mouse at different stages, you can always go to this web page. Yeah, so what Martin is showing, so this is a very good expression with very less background.
Here, for example.
Yeah, so the blue staining is what, that is the expression.
Um,
So as you can see here that there is quite some background. So it might be difficult to differentiate the actual stain from the background.
Also note that here is the sequence of the mRNA.
Yeah, but can you go below? If you can scroll down.
No primers for
Yeah. So this is because, so if you see that there are no primers listed, that means that this probe was designed using cloning method, not via PCR but via cloning. So actually they cloned the whole mRNA, and then they purified the clone. That's a very tedious work, and like people have stopped designing probes via cloning because it also takes a lot of time. So the easiest and the most convenient way is to design the primers and then do PCR. Martin, maybe you can also show the Allen brain atlas.
So there is a question from David. Can you use isolate signal intensity between two conditions for assessment of expression level differences? So between control and ischemia, for example. Yes, it's shown in the monkey niche database. You can do that.
Yes. So Martin, maybe you can open the monkey niche and we can show them, then it's more easy to explain. Yeah, so maybe you can explain some, like you can take one gene, which has a...
But I cannot open the slides with my internet. It is blocked.
Okay, then let me try. I can do that.
The important step when you do controlling ischemia is that the controlling ischemia should be done on one run. So the background is equal to both of them.
Okay, so I share my screen. Let's try. Okay, so I have here GFAP, which is the control. So here, so you can see, right? Correct. Yeah.
And you can compare the two images, the controlling ischemia in one window. So by clicking these windows here.
Yes. So here you can see, okay, I will zoom in. So this is the expression here, the SVZ.
And you can see the clear difference between the ischemia and the controller. So this is one of the genes that is mostly expressed after the ischemia.
So this is the control and this is the ischemia. So you can compare the expression levels and then you can say that it's high. What is the difference?
Yes, so I understand this. So you do have here also signal in a different topographic region. For instance, not only in the subventricular zone, but also, what is it, striatum or?
Yes, striatum.
Okay, so my question was, for instance, if you do have only the ependymal zone, like this, and you do have this substrate that produces, let's say, this dark bluish, what is it, signal.
Yeah.
And then it reaches a certain grade of saturation.
Yes.
And you do have the same in the control, and would like to compare this with your other condition. So would you then try to not develop that longer, or would you try to, what would be the strategy then?
Okay, yeah. So basically, that is why when I do the ISH, let's say, for example, I check the slides every 10 minutes or every 5 minutes, I keep checking the slides under the microscope. And once I see that I got enough signal as compared to the background, and there is, like, it has still not reached the saturation point, I stop the reaction using PBS or Milli-Q water. And at the same time, I have to stop the reaction also for the slide which we are comparing. So for both, for the control and let's say for example here ischemia, the staining time should be the same. So you have to monitor the slides every 5 to 10 minutes, and that way you can know like when you want to stop and not saturate the signal. Of course, like, and this ISH is qualitative and it's not quantitative. So you also have to keep in mind that of course you cannot like count the cells or the signal in the signal and compare.
Why? So we have self-detect and you can compare self-detect and other softwares that you can compare them.
Yes, you can use cell detect, but so for example, I have never used cell detect, so I don't know how accurate is that.
The important thing here is that when you compare two tissues or two conditions, the slide should be done at one go
Yes.
because of the background and the staining conditions. They should be the same.
Okay.
Dr. Pavlov, do you want to add something about because you are an expert in this field?
Not very robust and reproducible, though. I mean, if you'd like to count something, as this suggests, Martin, so you would go, let's say, into the one striatum and then into the other one, and then you would count just the number of positive cells, right? This is what you meant.
No, by signal intensity, it is measured.
Ah, okay.
So there are different strategies. It could be either by object number. I think this is what the cell detect does. It actually reduces the dimensionality. It reduces the resolution to a cell per pixel, and then counts positive cells in different counts, choosing different levels of saturation. Yeah, not exactly. There are many parameters there in self-detect, and the expert could tweak them. The other is if you don't have a saturated signal, you could actually use the levels, the intensity levels. And we have ideas that probably combination of intensity levels and spatial distributions of these intensity levels could be actually used for much accurate selection and counting, and probably also quantitation. But of course, this is there is no perfect solution because it would be used already. Our brain have a very interesting way to quantify them, but it is also very parameter rich. So basically, you need a very good expert to tweak the algorithm as well. And I think that many people are working on something to use more robust and more reproducible. But yeah, we are working too. So the main problem is that if some of the cells are already saturated, you basically cannot use them for quantitation there because you don't know above the signal how much is it, right? So we'll see.
Okay. But in this case now, in this present example, it's very obvious that the ventricular zone, I mean the ependymal zone, is much more intensively stained here on the left side.
Yeah.
Okay.
So the best way I think is to do the fish one with multiple genes, and then you can quantify and count them.
Yes.
But the scanning process is a little bit longer. So this is the page of Harlem Brain Atlas. They also have a section with Ice Age. With the number
So maybe if you could show there the data about the quantification.
Yeah, I'm going to do that. So for example, I want
energy, etc.
So this is the atlas. So they have many atlases, but this is the Ice Age atlas, and you can see here Ice Age cellular. And you can type the gene, for example, GFAP, and male brain. Those are all on mouse, I think. No, macaco mulata. Monkey, rhesus monkey. And you can see the staining at different levels. And they're saying that they're using anti-seize probe, of course. 48 months old monkey brain. And here is the lateral ventricle, so ventricular zone, of course. And as Pavel said, the expression, I don't know, I cannot explain this. Can you help me, please, Stoyan?
Okay, so can you combine ISH with, let's say,
maybe I don't know how are they doing the expression levels here on the membrane. But you have the option to do that, to compare.
Yeah, so like I have combined in situ with IHC. So yeah, so basically when you are performing, let's say, immunohistochemistry, it's always important that first you do the in situ RNA and then you perform the immunofluorescence or the immunohistochemistry. So yeah, although because in the in situ we are using protein escape, although that will degrade a few proteins. So I don't know like how this will work for the proteins which have a very low expression. But you should never perform immunohistochemistry first, and then because if you perform immunohistochemistry first, then you use protein escape for the in situ, it will already degrade some of the proteins, so you will lose a lot of signal then. And I'm not sure about trichromatic stain or eosin, so.
And the expression was inverted, right, Martin? So the signal was just inverted at Allen Brain.
Wait. I really don't know. I cannot comment on this.
Okay, okay.
I have load. I will send you the link.
Part, what was the protein that we have detected together with the in situ hybridization?
The proteins. If you have fish, for example, fluorescent in situ hybridization, you can combine 2 more proteins with the ISH. So the total amount is 3, and the only limit is your camera and your cubes on your microscope.
I meant because Path said that he has been trying to combine in situ hybridization with immunohistochemistry, and he's been trying, let's say, an antibody stain after the in situ hybridization. So my question was, what was the protein that you have tried to detect after the in situ hybridization? Was it a strong, abundant one?
You can, every protein should work if there is.
Yeah, so let's say for example,
there.
Yeah, I have used, let's say, for example, I did ISH on a long non-coding RNA first, and then on top of that, I performed immunofluorescence with PAC6 antibody or
PAC6.
TBR2 antibody, you know.
Okay, transcription factors. Okay.
Yeah, so mostly the transcription factors, and they all worked very nice because these transcription factors, they have a very high expression anyways. So even if some proteins are lost by protein escape treatment, I mean, that was not so, like it did not matter so much.
And was it fluorescence that you have been trying to do, or was it just light microscopy? What was it?
No, that was fluorescence. So both RNA ISH was fluorescent and also the immuno, and then immunofluorescence, yeah.
Cool.
I'm searching for some photos that I have on my computer so I can show you. So just wait a few minutes and I'll.
Sure, sure.
Okay, so now so I removed the slides from 60 degrees incubator, and we will again do 0.2 times sodium citrate wash for 2 minutes at room temperature.
I have found, but not my picture. This is from an article that is was published. Yeah, December. So transcriptome response and so yeah. But I remember that they have nice photos here, and you can see here triple staining. Here, GFAP, apelin, and DAPI. DAPI, so double staining, not triple staining. And you can see the signal. This is with fish fluorescent in situ hybridization. And this is triple staining. Here also double stain. Yeah.
Yes, I've forgotten this already. So the apelin was stained via in situ hybridization. GFAP was an antibody, right?
No, they're both ISH. I think so. Yeah, they're both ISH.
Okay, so there is no.
The BRD is antibody.
The BRDO is antibody and
the apelin is ISH.
mm,
I'm not sure about the GFP here. But the BRDU is 100% an antibody.
Cool.
Okay, so now we are done with the citrate washes. We proceed towards the Pbsd wash. So I've prepared Pbsd, and this is 0.1% 2 in 20 in PBS. So we do 2 washes for 15 to 20 minutes each. Okay, so now we have the second Pbsd wash again for 15 to 20 minutes.
There is a question from David.
Yes,
You have tried?
I'm reading it. Yeah, have you tried to analyze that?
Okay.
No, I have never performed ELISA with ISH RNA probes. Like if you want to do a quantification, then it's better to perform like, for example, single molecule RNA fish. Or as we discussed like before, you can also do cell detect. You use cell detect software to do the quantification. Okay, so once the Pbsd wash is completed, we have to lock the slides in the blocking reagent. So we are using 10% inactivated sheep serum in Pbsd. And this is mostly to prevent a high background stain. We will do this for half an hour. Okay, so now the blocking is completed, and now we have to add the secondary antibody. So here we have the anti big oxygen in alkaline phosphatase. The concentration we are going to use is 1 to 1000, and we will prepare 1 microliter of the antibody and 1 ml of sheep serum in Pbsd. So this will be our reagent. And again we will apply the antibody directly on top of the slide, and we will cover it with paraffin. So again, I will cover this with paraffin. We will incubate this for 90 minutes at room temperature.
I think an important note here is that there shouldn't be any bubbles beneath the paraffin. So when you are putting the paraffin, be careful when you to avoid forming bubbles.
So now let's say we meet again at 1 o'clock. So it will be
2 o'clock our time.
yes.
Okay.
Okay.
Any questions so far? Or part we can meet at 3 o'clock when the or 2:30. So at 3 o'clock there is the meeting with Christina Thaler, Dr. Thaler from the ICH.
Okay. So when do you want to meet? At 2:30?
Yeah, I think 2:30 is a good time for meeting. So
Okay, so
we can do it for 30 minutes, the rest of the protocol, and then we
yes.
can continue with the take-on.
Okay, yeah. So then 2:30 your time, we will meet.
Okay.
Okay. Okay. So once the hybridization is complete with the anti-deoxygenin, uh, we have to wash with Pbsd 2 times for 20 minutes each. This is to remove the extra unbound anti-deoxygenin so that we do not get a high background. So I already did the first wash for 20 minutes, and now we can do the second wash for another 20 minutes. And make sure that you use a very mild detergent for this wash. We do not want to use a harsh detergent to wash excess anti. So I use PBSD again, PBS with 0.1% to in 20. So after the 20 minutes of wash with PBS, we have to wash the slides with TMN buffer, which is basically again Tris NSCL buffer. And before before that, when we prepare the TMN buffer, first thing is it should always be freshly prepared, and we have to add levamisole hydrochloride powder. So the levamisole is basically a quencher. So it's a quenching reagent that will block any alkaline activity or any endogenous phosphate activity which is present inside the tissue. This will result in very less background. So I have already added levamisole here in the TMN buffer. So I will incubate the slides at room temperature. You can do it 1 time for 5 minutes, or you can also do it 2 times for 5 minutes. So meanwhile, while the slides are in the TMN buffer, we will prepare our staining substrate. For that, we will need NBT. So as you can see, this is the NBT, and we need BCIP. So this we have to add 1 microliter of NBT and 3.5 microliter of BCIP per ml of TMN buffer. So I will prepare this reagent now. And since this reagent has to be kept in dark, I will put an aluminum foil over the bottle so that so that the reagent is protected by light. So after 5 minutes, we will discard the TMN from the slides. And now we will add the TMN with the substrates NBT and BCIP. Now, because the whole reaction has to be carried out in the dark, either you can place the slides in a dark room or a box, or so what I will do here is I have an aluminum foil, so I will cover the slides with the aluminum foil. I will cover this box with this foil. So like this. So now the reaction can be in the dark, okay? So this is, now we have to keep this for more than an hour, but what we have to do every 10 to 15 minutes, we have to check the slides for the signal development. And if you feel that the signal has developed as you need, so before the signal reaches the saturation point, you have to stop the reaction by putting the slides in PBS. So you have to monitor the slides every 15 minutes. Now I will put the slides in a dark box. Okay, so now we have almost completed the manual in situ hybridization. So what I will do now is now we will give you a demonstration of our Tikan robotic ISS platform, and we will show you how do we perform in situ hybridization on a very large scale. So yeah, so we will do the demonstration in 5 minutes. Martin, can you see the robot?
Yes, very clearly. Maybe a little bit up. Your camera, I mean.
Martin,
Yes.
can you see the robot?
Yeah.
So now we are going to give you a demonstration of the ISH Tikan robot. So Mrs. Thaler, she's here and she will give you a demonstration of how the robot works. She's an expert and she has been working on this technology since quite many years now. So this is Mrs. Thaler, and now maybe you can start.
So basically, the advantage of doing the ISAC robotically is mainly that it allows you to do many samples at once. So you can also think. So you can also think about doing a comparison of various stages or types of tissues that otherwise it would be rather difficult to do in such a large number. So it's basically the same procedure that Bart has described before, but it's just that we can do this with the help of a pipetting robot. And basically we have a setup that pipettes sort of from a certain vessel to the slides. And the other main feature is that we are using a flow-through system. So that means that we have that the samples are basically in a flow-through chamber that allows us also to use sort of a minimum of reagents to some extent. And we explain?
Yeah.
Have you explained this?
No.
Okay, so the principle of the flow-through chamber is that the robot just pipettes a certain amount of liquid from a certain vessel to this flow-through chamber, and then the liquid flows in at the top into a reservoir, but by the time the reservoir is empty, liquid stays in there. So this allows us to do longer time incubations for the tissues to staying in place and being incubated for certain amounts of time as required for the protocol. But the basic system is just a flow-through system. And then, of course, you can wash, and you can add different reactions at specific times. Usually robots are more precise than humans are, but not only that, we also have sort of a temperature control in the sense that we have very powerful water bath that control the temperature of the hybridization chambers at any given time, sort of whether it be room temperature or usually we have it as at 60 to 62 degrees overnight, okay? So this is the main difference between doing the hybridization manually and robotically is that, of course, you have a short time to finish the protocol because you can have the robot do things overnight that you only want to come to the lab to do. But otherwise there's not. It's basically just a pipetting robot that's adjusted by the protocol to deliver the solutions to certain hybridization chambers at certain times in the required amount, and that's pretty much it. Okay?
Okay, so I will explain.
So, are there any questions?
Mark, can you show the chambers more up close, please?
This one?
The chambers, the glass chambers.
Okay.
Okay, so those are chambers to hybridize.
So these are the chambers. Okay, so this is
Yeah, but maybe
glass.
it's better if.
Yeah. Okay. So let me. Yeah. So we will show you how do we assemble the chambers now.
Yeah, it will be great.
Yeah, so here you can see just on the question of it. Yeah. So this is the metallic plate which we put first on this rack. No, there. Yeah, it's perfect. Yeah. And then on top of that, we put our glass slide with the tissue sections. Yeah, and then we have to space. So we have to place a 1 millimeter of spacer. So now we have to put a spacer which is like 1 millimeter, and then we cover the slide with a glass chamber. So it's a relatively thick chamber. Maybe Krishna, can you show that chamber once? Yeah. So now we create. Yes. And we have to clamp the whole setup. So the first clamp at the top and the second clamp at the bottom.
Okay.
Yeah. Yeah, and then we put the whole setup here.
So basically, it's really the right angle
Yes.
so that the flow through is.
Okay, and what is the maximum capacity that the robot can do at once?
So the robot can handle 96 slides at once. So here, there are two racks. So the first rack can handle 48 slides and the other one also 48 slides. So in total 96 slides. So now you see that the robot, so the pipettes are pipetting the solutions.
How often should you visually inspect if the robot is sucking liquid?
So there's a question that how often do we check that the pipettes are taking the solutions in the correct amount?
I mean, this is what we established at first. I mean,
Yeah.
of course, the flow through is the same, and then, I mean, the basic problems might be air bubbles.
Okay, so let's say, so there are 8 pipettes here. So, okay, and if one of the pipettes or if a few pipettes do not pipette the solution correctly, then we will get notified on the computer. So we have the computer which is connected to the machine, and so there's a script which is running, and the script will show us that if some pipettes have not pipetted the solutions correctly. So this is how we know that everything is working fine.
It's actually more precise than any human one.
Yes. And basically, if there are solutions which are not placed properly, or if there are solutions which are empty in these chambers, then also the software will notify us that the solution is missing. So it will not pipette just empty. So you see now. So it's pipetting one of the solutions, and
That's
now it will go. So there is also a system that the pipettes are washed after every solution. So they are washed with milky water. So now you see, now it's washing the pipettes automatically. so that there is no cross-contamination between solutions.
One more question.
Yeah.
What if the robot introduce a bubble inside the spacer in the chamber, I mean?
Yes. So there can be bubbles which we have to often check. So we have to check individual slides from time to time and see if there are any bubbles formed. And if there are some bubbles, we can just tap it out.
Yeah, but usually we don't really have major problems with bubbles. I mean, what we use to avoid this is that we add tween to all the solutions so that the flow very evenly throughout the hybridization chambers.
Yeah.
Okay?
Yeah. So the hybridization is also carried out in these ones, and these racks can be heated up to 60 or 65 degrees for the hybridization to be carried out.
Does the robot pipette the riboprobes by itself, or should the riboprobes be introduced by the researcher?
So I didn't quite understand.
Yeah. So he's asking that. So does the robot pipette the probes by itself, or do we have to pipette the probes and anti-dig?
Everything that needs to be pipetted, the robot can do. You just have to put the solutions in the appropriate chambers, and then everything else runs automatically. So this is why basically we can have the results within 24 hours.
Yeah. So whenever there is some error, or we have to or a solution is missing, so as you can see that either the robot will beep or it will show a flashing light like that. So like that. So now it's asking for a solution to be pipetted because the robot recognized that there is a solution which is missing. So it's flashing. Yeah.
What else can you do with this robot? Can you do immunohistochemistry?
Yes, we can. Yeah, so we have to first write the script for the robot, and yeah, technically you can do immunohistochemistry, immunofluorescence, or maybe single molecule FISH on the robot. Yeah, of course.
Anything you have many pipetting steps.
Yes. So basically, anything where there are many pipetting steps, you can do with the robot.
What about some other, for example, cell cultures? Is it well adapted for cell cultures? Can you adapt it for cell cultures?
No, we cannot do cell culture work here. We cannot do cell culture work here. So he's asking that can we do cell culture?
Cell culture, not in here, but there are different setups. It would allow you. Because there are setups for this plates, basically tissue culture plates and so on. But I mean, this is in a different program and so on. Okay?
Yeah. So not this one, but there are different versions.
So you can buy an additional equipment and you can modify this, for example, this one to do the cell cultures.
Yes.
Or you can buy another robot just for cell cultures.
Yes.
One more question. Which is cheaper, the manual in situ hybridization or the robotic one?
Well, I mean, I would say there's a certain limit to what you can do manually. And it's not just a question of what is cheaper. Of course, to some extent, it's much cheaper if the robot does it, because we can use very small amounts of relatively expensive reagents. But it's also a question of sort of how reliable and how, no human is as reliable as a robot. Okay? The robot, you tell him, okay, he adds a certain amount of solution, to a certain space at any time. And the advantage is, of course, that so we start usually in the afternoon of one day, and we have the results by noon the next day. I don't know if that answers your question. And also you can, I mean, you know, even on this small robot, we can do almost 200 slides a day. There's larger robots that can do 400.
So of course, when you're doing such a big database, you need a lot of slides per day done. And manually, you cannot do that many slides.
Yeah. So so he's asking that when we want to create a like a database, then this robot is much useful.
Yeah, of course. I mean, if you look at something like the Allen Brain Atlas, I mean, this was created by this technology, okay? I don't know.
Yes.
So
We have
that,
already talked about the Allen Brain Atlas and the
Okay.
Pipette, so we
Okay.
showed them how it was established.
so this is really how we did all this. Otherwise, it wouldn't have been possible. And then I think it's also a question of, you know, reproducibility. Of course, I mean, you know, if you do just a few slides, you try to be as reproducible as possible. But if you do hundreds and hundreds of slides, you cannot have the same.
Okay, thank you for your
Okay,
answer.
I hope this answers your questions. I mean, if you have more, just please feel free to ask.
Is it hard to learn to do the robotic ISH, and how much time does it do you need to learn to do the robotic?
Yeah. So I can answer this because I have recently learned. So it's not about actually learning. So the learning how to operate the robot is basically very easy. You just have to put in solutions together and be careful that everything is working fine. But the main thing is practice. Like, you know, there are so many critical steps in between that, and the robot is very sensitive. So even if you miss a small step somewhere, it will shut, like, so it will crash, you know? And it's basically about how many times you practice.
Yeah, that's why we use very strict protocols. You know, it's basically, it's all controlled by basically checklists, if you like. So you have to, you know, before you have you done this, check, have you done that, check, and so on. So that's really how it works.
And
Yeah.
of course, the preparation of the liquids prior to the ISH, the robotic ISH is
That's also very important.
Yes.
How do you prepare the tissue? And yeah, that's extremely important. And it depends on whatever you want, sort of in a specific case. What do you want to see? What do you want to visualize?
Mm-hmm.
Okay.
Yeah.
Bart, can you show, is there a list of ingredients that you use, that you put on the robot so you can check them? You can show the participants the file.
Do you want me to send a PDF file? Then that will be convenient.
Oh, okay, no problem.
Okay, so I will send a PDF file.
Okay.
That's better.
And I
No,
will,
but in terms of different types of tissue, and so I mean, I've been working for a long time on this robot, and I mean, we've done anything, you know, any tissue imaginable, from human to chicken to mouse, of course, rat, you name it. And so you always can do it. And you can also do it on paraffin sections. You can do it on relatively old formaldehyde process sections. I mean, if a probe is really good, then you can still see the signal.
Yes.
So it's very versatile. I mean, it's, yeah, whatever you want to do.
Yeah.
And when you introduce the MBT and BCP compounds, how do you check the slides for staining? Because you said
Okay,
that.
so this is a critical point. What we usually do is that while the color develops, you check certain slides and you see, okay, so yeah, this is just about right. And then you can stop the reaction.
So basically we have to check, like we have to take out every slide from here. And so if you turn around the slide, so you can look at the expression under the microscope. So there's a possibility you take out every slide and check it.
While the colors develop.
Yes.
So you can say, okay, yeah, this is fine now, so I stop it.
Yeah, and for example, yeah, so if the expression is not yet developed in this slide, you put the slide
You
back,
don't.
and if it's done, then you separate out the slide and stop the reaction.
Do you inspect the slide under the microscope or just visually?
Yes, under the microscope. So we have a Sarah microscope right next to the robot, and then we just check. And, you know, to some extent, it's a question of experience when you can say, oh, yeah, this is fine.
But when you have 96 slides in there, it is very difficult to see all of
Yeah,
them.
but usually, sort of, not each of the 96 slides come from a different gene. Usually, if you have 96 slides, then it's like a certain number of slides that belong to the same gene. So you take at least one, and then you see. And of course, I mean, it's never like from when you do this the very first time, you know. So at first, you also have the question is how good your probe is, which means that, okay, if you have exactly the best probe that's possible for this gene, which sometimes also, you know, it takes a little bit of fiddling around to find out what this is. But, you know, once you get it to know, you know exactly. Okay?
What about positive and negative controls? Do you include those also?
Yes. So what negative controls do we include?
Okay, so we always have positive control, negative control. So we have negative control is basically you don't want to see any background, yeah? And positive control is a gene that we know for sure is expressed in the region of interest.
So for example, in our lab, we use neuropeptide Y, NPY, always as a positive control.
Yeah, because
it
they're
always
widely expressed.
it's a housekeeper gene. And for negative control, either you can use a sense probe, or you can, or you
Just
do not
have
use
a
any
blank.
probe. Yeah, you just apply the hybridization buffer without any probe.
Okay. Thank you for your answers. It was very helpful.
Okay, so if you have more questions, just contact us, okay?
Yeah,
Okay.
thank you.
So now I will ask Murad if he is joining.
Oh,
Okay. He's not picking up my phone, so maybe he's busy.
Okay. Meanwhile, you can show us one or two slides under the microscope.
Okay, yeah, do you have some?
Yeah, but I don't think it will be possible to see under the microscope.
Oh, how to visualize.
Yeah,
Maybe can we,
I can send the pictures. That will, because how should I put the camera on the microscope?
Okay.
Yeah.
yeah, exactly. It's a question of the camera. And
Yes.
yeah. But I mean, have you looked at the brain, at the brain atlas and gene paint?
Yeah.
But yeah, okay. But I want to tell you one thing. So if you just look at the overall picture that you get usually, it's relatively small, and that doesn't give you all the detail. So if you have to really enlarge the images so that you see that yes, there's real cellular resolution. Okay.
Mhm.
So I mean, because I've seen many instances where people just look at it very superficially, and then they don't get to see where things really are expressed or not.
Yeah. Thank you for the advice.
So it's very important to really study that.
Okay.
And it also depends on what you want to see, what you want to know. Okay?
Mhm. Yeah.
But
Thank
if
you.
you have any more questions, just feel free to ask. Okay?
Thank you.
You're welcome.
Can you hear me now?
Yes, now I can hear you. Hi.
So we are waiting for Murat to join after 1 hour. By the way, what are you cutting?
Brains. But not my brain. Still.
Okay, I will. Maybe I will call you when we begin.
All right.
But I don't know if I have your number.
So what are we looking at now, right now?
This is the Robotic H. So this is the robot. This is the beast. So you can see the pipettes there, and the black things are the spaces where you can put the slides.
Mm-hmm.
The total amount of the slides is 96. So you can do 96 slides for 2 days. So the protocol is approximately 2 days.
Crazy.
And the amount of the solution that you are going to use is a lot smaller than that we use in the manual ICH. Because the glasses I'll show you now, let me just find it.
Okay, I'll rejoin then in approximately.
Wait a second. just to show you the piece of glasses that they are using. They're very interesting because they have a very shallow groove that can 10 to 15 milliliters of liquid or something like that.
Mm-hmm.
I cannot. Ah, I find it. Can you see?
So now I can see. Oh,
So this is the robot, and this is the glass lights. So you have a glass with your specimen, a spacer, which is a rubber thing that prevents touching of your specimens and the glass. And you can see here is the glass, which is very thick, by the way. It's thick, like that cell phone. And it has a groove here on the top, and the robot pipettes a liquid and puts it in here, and the liquid disperses very slowly through the specimen.
uh-huh. Whoa.
Which is very cool.
This is my robot. Yeah.
Okay, please give me your number here on the chat so I can have you in my contacts, and I will call you or write you when we begin.
All right, just let me find the logo. Oh. Okay, I'll write it.
Okay. Have a nice cutting.
Thank you.
Did you cut all the tissues? Where
It's
are the
here.
buttons?
Yes.
Connecting, and we will start shortly. First, to introduce you, Murat has obtained his PhD in Max Planck in Hanover, where he worked in robotics, but was doing a lot of robotic ISH. He has worked since then in many pharmaceutical companies, including Abbott, Genentech, and again in Max Planck Institute for Biophysical Chemistry in Göttingen. And now he's a senior scientist at a company called Archer DX. And we will have now a discussion with him, some troubleshooting. We have questions that ask during those 3 days. He, Pat, and I will answer them.
Oh, there we are. I was muted.
Hello, Murat.
Hi, Martin. How are you doing?
I'm excited. I'm fine. Thank you.
Very good. Very good. So, shall we start with the first question right away?
Yeah, let me just share the questions so everybody can see them.
Sure.
I think this will be the best.
Way to go about it, huh? If you want, I can read the question first.
Okay. I am sharing now.
So, oh, excellent, excellent. So what I did is I kind of like sent out the answers to Martin. So you have the questions on the screen now. And right below the, the questions, you have very brief answers. So for example, like how stable is TAC if rethawed multiple times? So my answer there is the DNTPs are more sensitive compared to the TAC. TAC is a bit more stable, but definitely, multiple times thawing for the TAC is pretty bad too. But the first thing that will go will be your DNTPs. So, especially the DNTPs, you need to make sure that they're carefully aliquoted so that you don't thaw them multiple times and that you can just take an aliquot to prepare the PCR mix you're going to prepare at the time. So second question, is it possible to prepare master mix? Yeah, of course, you can do this. In fact, most of the new TACs are sold in 2 times mixes. So basically, they're more dilute. And again, just like the DNTPs, if you aliquot those out into single-use tubes, you can just thaw one out, use it for your PCR for the day. That way you'll prevent multiple thawing. And what else do we have? How do you know what your expression is? So, okay, this is a bit of a long question, but basically, if you Google NCBI gene, you'll go to the web page, which I can't show you because Martin's sharing now. I'll show that to you at the end. So Martin will stop sharing, and I'll show you the web page. This is basically.
Yeah,
don't worry about it for now, Martin. We'll go to gene on NCBI, and then I'll show you how to look up a gene, your gene of interest. And there, underneath the gene of interest, you have expression. Oh, you removed it? Okay, let me share now. Here, I'll share. Here we go. We'll share this. Okay, there we are. So if we go to NCBI gene, so this is the web page. It's ncbi.nlm.nih.gov/gene. And my gene of interest here is VIP. So this is actually vasoactive intestinal polypeptide. This gene actually marks interneurons. And if you go down here, you can see the expression for this in mouse. If you, of course, pull up the human version, so if you say human VIP, it'll give you the expression in the human too. So this is a gene that's in interneurons in the brain, so that's why it's coming up in the cortex of the adult very high. But as you can see, it's in intestines in the adult. And let's see if it's in the lung. Oh, it's not very, very high in the lung. Interestingly, a Swiss company just made billions from this polypeptide, by the way, because they realized it decreased congestion in COVID patients. Anyway, so next question. Any specific requirements concerning the tube materials and low bonding wall thickness? Well, what you need to do here is you need to mainly match your equipment. So you need to get the right commodity, the light plastic, that's suitable for your equipment. Now, what does this bring up? This brings up the question, if you don't have a very highly advanced PCR machine that uses thin-walled PCR tubes, you might need to adjust the conditions. You might need to increase the, the 95-degree denaturation step, the annealing steps, or the elongation steps. You might need to play around with it, the timing on your cycle, so be careful about that. And let's go to the next question. If we do not have a PCR machine with controllable temperatures, what shall I do? Very easy. Just go for the midpoint. Just take 55 degrees. Normally, you could do a gradient from 50 to 60. So then see what comes out at 55, and depending upon the result, if you get a band that's like, weak, but with a smear, start increasing the temperature, you know, and unfortunately you're going to have to do it multiple times. Another thing is you can just keep the temperature stable, but you can change other parameters like magnesium chloride. Just change the concentration of magnesium chloride, do it at 55, see what happens, move on from there. Should I use a positive control? You know, absolutely, but don't forget your negative control is more important, because if something comes up in your negative control, that means your reagents are contaminated. So what you would have to do at that point is you need to refresh them. Basically, you might need to throw them out, and you can check this one by one. First, check by changing your aliquot of DNTP. You know, go on from there. Make sure that your negative control is negative. Negative control is basically all your PCR reagents, you just don't put the cDNA. So if something comes up in that, you're in trouble, because that means one of your other reagents is somehow contaminated with either a PCR product or with a foreign cDNA. Your positive control is important, and Martin can probably share with you guys a positive control. He probably has a PCR that works. Just grab that. If he gives you primers, you know, that's fantastic. If he doesn't give you an aliquot, just design your own primers, use that as your positive control. If your first PCR works, then you don't have any problem anyway. That's your PCR control. Should I be concerned with too much primer dimers? Well, you know, if you do a gradient, this shouldn't be a problem. But most importantly, primer dimers are very small. So when you do PCR purification, like with columns or with gel purification, you don't need to worry because you're extracting a specific band. If you're using a column, the column will preferentially extract the larger size sample. So generally you lose most of the primer dimers. So again, that's not too much of a problem at that point. So let's move on. What do I do on the gel if there are more than one bands? Do I just, so whoever asked this question is already answering. Basically you would do gel purification, just exercise the band from the gel, and you would just re-amplify that. Another thing to do is you can change your gradient conditions. So sometimes when you change the condition, you get more stringent, and at that point it turns into one band. Another thing might be, it might be real. So there might be an isoform for your gene. Under those circumstances, it shouldn't be a problem unless you're interested in a specific isoform. Anyway, moving on again. If on temperature gradient PCR, all the bands look the same, what do I do? That's actually very easy. That's actually a good result. If they look all the same, and if it's the right size you want, you don't need to do anything. If it isn't the right size you want, that's actually a very big problem. Then just design new primers, because those primers are somehow working pretty bad. Just switch to the new primers and everything should be fine. Instead of a clear band, I have a smear. Well, that's really bad. One reason might be that your cDNA might be bad, or the temperature using for the gradient might be very bad. Or like I said again, for the previous example, you might need to design new primers. It might be non-specifically binding to another gene in the same family, or that's in a kind of like an associated family gene member. And that might give you a smear. So to actually get a specific product, just design new primers. A smart way to design your new primers is you can do something like a nested design. If you design them outside of your previous primers, you can see if that works. If it still gives you a smear, then you can use the old primers to do a nested PCR. So basically you use the outside primers, then the inside primers, and you do a nested design, and that will hopefully give you a good result. What is the shelf life for purification columns? This is actually, those things, they're basically like they survive forever. What you're more worried about is the solutions, the liquid components. And if you carefully store those, they should last for a very long time too. I mean, when I went to the Max Planck, the Max Planck is a very controlled environment, though, very nicely air conditioned, and everything stays in pretty good shape. So years later, when I visited as a guest scientist or as a postdoc, the stuff that was there from my PhD was still working. Just to give you an idea, guys. What is? Here we go. Wait, what about different Taq polymerases? What is your experience? So basically, different Taqs can be an issue. This really does make a big difference. You might need to optimize all your conditions if you change Taqs. Like we use Quiagen, everything that we've done has been established with the Quiagen. No, wait. Yeah, it is Quiagen. Sorry. Yeah, the Quiagen Taq polymerase. We're very used to optimizing our conditions with that, and we're very happy with it. So if you do change it, you're going to have to re-optimize everything. You would have to re-optimize the primer concentration, your PCR conditions. You might need to optimize a lot of things. Fundamentally, they should be similar. But, depending upon if you were using something like a hot start or a high fidelity polymerase, things can really change. So number 13 is a bit of a long question. What I would recommend is here, actually, you guys can see my thing, right? My screen. So write down my email. So whoever wrote this question, contact me by email. Thanks, Martin. Just, contact me by email. I'll examine the paper and I'll get back to you. I'm hoping this question was associated with this second question, but I'll get back to this question later on. Basically, I need to figure out what type of probes they're using. There are a lot of papers where people kind of use in situ hybridization with LNAs and stuff, where they target mutations and different, karyotypes and whatnot. But let me examine the paper. I'm going to get back to you on this one. Basically, question 14 is, it's about using ISH with different methods. I have already used it with a lot of other methods, one of them being IHC. So doing either protein detection first with an antibody, and then, comparing where the transcript comes out, especially for proteins that are released, this is different because they're kind of like produced in one cell and they can be kind of like flowing somewhere else and released in, a different part of the cell body, like, for example, synaptic proteins and stuff like that. Those can make a big difference. And you can also, do it together with other methods. The only thing you need to watch out for is that you do not ruin what you're targeting in one process compared to the other. For example, like if you're doing ISH and then you want to do something like chromosomal localization, after you detect with ISH, you would probably have to pre-treat more with pepsins and stuff before you can detect and do something like a chromosomal localization or something. The same would go for ISH and IHC. If you destroy your protein or your RNA while you're doing one process, you won't be able to carry out the second process. So make sure your process is streamlined properly. And you might need to switch. Like if you try IHC and then do ISH and it doesn't work, try ISH first and then try to do IHC, and it might work that time. How expensive is the product? Yeah, this is a big problem, especially if you're using non-radioactive ISH and you're using tyramide signal amplification. It's pretty expensive. If you're using a robot on top of that, it's even more expensive. If you ruin one run, you could probably basically go on holiday with that. And so it's very expensive. It's around like $10 to $20, depending upon which country you're doing it in. And my suggestion here is you could just switch to conventional radioactive ISH. That's with like cheap radioactive isotopes. Of course, you might die of cancer, but that's fine because it's cheap. So Anyway, after the incubation with my RNA probe on my slides are bone dry. Why? Okay, so this is an interesting question. So what's probably happening here is sometimes the labs are too dry, and if you don't carefully moisten the environment that you're carrying out the hybridization, what can happen is you can get a lot of evaporation. Make sure that you really moisten the environment around the hybridization chamber. And you know, if you do that, the chance that it will dry would be very low. The other thing is, if you're doing it overnight, cut down on the time. A lot of the modern procedures have shown that, you know, doing it for about 2 hours is enough. You might be able to finish it off in 1 day. You know, you need to test these. If you can't do it that short, my suggestion is maybe doing another addition. This is very easy with a robot, but of course, you know, coming in the middle of the night, adding more probe to your slides that are about to dry is very difficult when you're doing it manually. So with that, you just need to make sure that the environment, the hybridization chamber, is moistened very carefully. And if you do that with stringency wash solutions, you know, the low stringent ones like 5 times SSE and stuff like that, you should not have any problem. And that's a very cheap solution. So it's very easy to moisten your chamber with that. And if it accidentally interacts with your probe, it won't wash off the signal because it's low stringency. Yeah, basically that's the workaround for that. That's the best thing you can do. Okay, guys. So that's all for now from me. Do you guys have any other questions? Yeah. You're very welcome, Martin.
Hey, Murat, how are you?
I'm fine. How are you, Anton?
Good. And I'm happy to see that you are doing good too. What we can do is we are planning a follow-up of this course.
Yes.
And there you maybe could present the multiple fluorescence paper. Like where you combine 5 or 6 different fluorochromes at different wavelengths.
Right. So we haven't published that, but what I can definitely present the method if you would like. That's not a problem. We can definitely do that.
So I would like briefly to say, and because I saw I had a glimpse on the manuscript. So they were able to detect up to 6 different wavelengths on one and the same tissue section, right?
Yes. So, by the way, Parth's here too. Excellent. So one thing I realized for that is, so when we were doing too many multiplexings, one of the things that happened during the process is that the tissue, because we were using fresh frozen, initially, when I developed the method, I used FFPE in America at Genentech. So we were trying it on fresh frozen over at the Max Planck. So after 2-3 tries, the tissue started disintegrating. However, later on, after I returned to America, I realized what the troubleshooting for that was. What you do is basically you keep your not the FFPE, but the fresh frozen tissue in PFA overnight. So you cross-link it like crazy. So you basically turn it into an FFP-like structure, and then easily you can do it up to 6. But, you know, this is something I haven't tested. We would have to test that. But it works with FFP. You can easily do it up to 4-5 channels with FFP, and that's what I will present to you guys. Because I'm sure you guys can easily work with FFP. You guys must have tons of FFP tissue there.
Yeah. Thank you.
Excellent.
Any other questions? Anyone? So our consultants.
Okay, guys. So this is actually pretty good. It only took 20 minutes.
You are happy in the morning to have no more questions. Did you get your coffee already?
Yeah, I already had my coffee. I mean, I can't do anything without drinking coffee. I actually took my cup downstairs, but yeah.
Okay, anything from your part to add?
No, so far I have covered everything. So most of the questions also we have like answered, and if they have more questions in the future, they can always contact us.
Absolutely. Yeah. And so I'm going to say goodbye, guys. Take care. And like I said, I gave you guys my email. Just reach out to me if you have questions, especially the person that sent out the manuscript. If he reaches out to me, I'll give them a reply later on. Okay?
Yes.
Yeah,
Thank you.
I will tell her.
Yeah.
So, anything else from David? Questions, comments?
I do have one question. So first of all, thank you very much. It was a fabulous workshop. Thank you, Murat, for all the comments. Very nice to hear the answers. And I do have one last question. Have you ever tried to do in situ hybridization on EPON, on resin-embedded slices?
On resin-embedded slices? Not me. No. I don't know if anybody else.
Possible? What do you think?
Yeah, absolutely. I mean, so David, it should be possible. So first of all, you know, the only problem there is you need to optimize the conditions. So FFPE tissue, I used to call tissue with fossil RNA, basically. The reason is, like, you know, some of it can be treated so badly, like it's been formalin fixed so bad, to recover it really is very troublesome. But if you kind of generated a way to, you know, expose the RNA, you should be able to do that. What you might want to do is, so the multiplexing that Anton mentioned, I did with branching DNA. So the chance that your resin contains fragmented RNA is highly likely. So what you can do is you can either use the old trick from whole mounts, whole mount in situ hybridizations, or you can use the modern technology, which is branching DNA, to try to access the RNA fragments and get a picture. The branching DNA technology works great because you can have different branches. In the whole mounts, you basically take a long fragment and you separate it into small parts. But that actually is a very good question. I love that question, by the way. I mean, so yeah, you should definitely try that. I mean, I certainly believe it's doable. I would presume it's preserved in the resin, but you would have to give it a try. And one of the best things to do that would be create an artificial resin embedding strategy, embed a lot of samples that you know contain the RNA, and try to recover it from the resin embedding, and then you can try to apply those same conditions to the resin sample that you're going to work with in the future. So, you will have to optimize the conditions, that's for sure.
Cool. Gives me a lot of hope for Christmas.
Excellent. It should
Yeah, the multiple fluorescent signal is like a Christmas tree.
Okay. So,
anyone else of the other participants want to ask something?
I think we're done. Okay.
Yeah, so I would like to thank very much
Thank you for inviting me.
Yeah, this quick entrance, solving all the problems, and then quickly disappearing for the rest of the coffee. Thanks so much, and then we keep in touch, and we see when whether you can really show this thing about the fluorescence.
Sure. No problem at all.
Thank you.
Absolutely. Take care, guys. Bye-bye.
Bye.
Bye.
Bye. Okay, so we're coming to a closure, I guess. So I would like to again invite Dr. Parth Joshi, who was the main strength behind this workshop, to say a few words. Thank you, Parth. It was excellently prepared. Of course, thanks to the expertise of the department of Professor Eichele. But you did a great job, I think.
Yeah, thank you very much, everyone, for participating. Like, it was a very nice workshop. Everything went very smoothly, so it was not a problem. So
yeah, we are already looking for the second part related to the fluorescence passage.
Yeah.
So we keep in touch. I believe we are going to make this available, the videos of these days available on YouTube when if the YouTube doesn't crash again. But you'll be able to see that. So I guess this is a unique course in Bulgaria for the first time. Also thanks to the national program, European Research Networks of the Ministry of Education and Science. I would like to thank them again for their support. And we are going to extend this course then with the second part as well in the future to come.
Sure.
Yeah, so thanks everyone participating.
Yes.
Thank you, Ministry of Education and Science, and we are looking forward to the next events.
Okay.
Bye-bye.
Bye.